Okay, it's recording now. We're still speaking about DNA code being used in order to make a protein. Now, the sequence of bases there is going to be used to form messenger RNA. That's what we've been covering. So I'm going to use this space over here. So what we've been covering in terms of protein synthesis, we've been saying that in order for us to make proteins, we start by using a code from DNA. So a code from DNA whereby a section of DNA that codes for a certain protein or codes for making protein is going to unwind. And then when it unwinds, the bonds are going to break and the molecule will unzip. Then free RNA nucleotides are going to be used to then form the messenger RNA. That's what needs to happen, right? Then from the messenger RNA, this messenger RNA will then move out of the nucleus and then attach itself to the ribosome. In order for transfer RNA to be formed, it will use the codons from the messenger RNA in order to have the specific amino acids that are coded for by the anticodons. So we start with DNA, and then we have messenger RNA, and then we get transfer RNA. The transfer RNA will then pick up specific amino acids that are determined by the codon of the messenger RNA, which is complemented by the anticodon of the transfer RNA. Then we form the amino acids. The amino acids are going to be taken rather from the cytoplasm. So we don't make the amino acid, they are already available in the cytoplasm. The transfer RNA will pick up the specific amino acid, bring it to the ribosome, pick up the specific amino acid, bring it to the ribosome, pick up, that's all it does. You then end up having a chain which is made up of minimum 50 amino acids, and that chain that has a minimum of 50 amino acid, we then call it a protein. Now this is the goal. And we would have achieved our goal if we used the genetic code from DNA and then we make a protein. Happy. Now, during this whole process from DNA to messenger RNA, transfer RNA, and amino acid, it is possible for a certain change to occur. And that change that can occur, we call it mutation. Now, most of the time, we speak about gene mutation, which is what we're going to speak about now. Then when we do meiosis, we are going to speak about chromosomal mutation. So to break it down, gene mutation speaks about a change that may happen in the sequence of bases, because remember, a gene is a section in DNA that codes for a certain characteristic. That section is determined by the sequence of bases. Now, this change can literally be caused by anything. So if, for example, we have bases TAC, and these are the bases that we have. I'll start from DNA. I'll just write it here. In DNA, we have TAC. For these bases, if we are forming the messenger RNA, the basis of the messenger RNA or the codon will be AUG, meaning the anticodon that is going to be formed here will be UAC. I hope I'm not moving too fast for people. This is what we've done before. Then this anticodon will then determine the specific amino acid that will be collected by the transfer RNA in the cytoplasm. Now, I'm just going to give a name of the amino acid that's coming to my mind. The name of the amino acid will be proline for this particular base. Remember, I'm doing for only one triplet, and there is a long sequence of bases in DNA. So for this particular amino acid that we formed now, it is proline. Then obviously maybe another amino acid can be arginine, another amino acid, trypan, whatever. Now, if there is a certain change during DNA bases being used to form messenger RNA, a change could be, for example, C being replaced by G. In this particular triplet, where we formed amino acid proline. So where now C is replaced by base G. So there was nothing that changed with T, nothing that changed with A, but we initially had the base C. But in that specific position, now the base C is substituted by base G. It was C here. I'm not sure why it looks like a G now. So this was C. Yes, it was C. Yeah, so that becomes clear. So now I want us to look at what has happened here. So we are still speaking about the same triplet, but what has been the change now? We initially had TAC, but now we are having TAG. G base has replaced the C base. This is the change that happened in that triplet. Now, I want you to look at this. If there is a change that occurs in DNA, one way or the other, it will have an impact on messenger RNA, transfer RNA, amino acid, and protein. If you mess up your ingredients with your scones, if you're baking your scones today and you've measured everything and it comes out perfect, yay. But then again, you're making the scones again, then you put too much salt and then you forget to put sugar, the scones are going to taste different. So this is exactly what is happening. There was a change that occurred in DNA, and this change can result in changing the protein that has been formed. Let's trace it back now. So if there is a change that occurred in DNA, what will be our codon in messenger RNA? We are going to follow this. Remember, we initially followed what we had, which was TAC. And now there was mutation that occurred. We are now having TAG, meaning the codons for that specific triplet will now be A, U, C. I hope you can see the change. We initially had what? AUG as our codon in messenger RNA. Remember now I'm using terminology codon. Those are 3 bases in a messenger RNA strand. And then we are now having AUC. What am I using to get AUC? I'm using this TAG, so I'm moving like this. Then from AUC, I need us to get transfer RNA. We are going to use AUC to get transfer RNA. A will be paired with U will be paired with A, and C will be paired with G. So for now, for transfer RNA, what do we have? We have UAG. Now, there's 2 possibilities here once we get to the amino acid. So I'm sure you can trace what is happening. There was a change in a DNA base, which led to a change in messenger RNA, which led to a change in transfer RNA, because initially our transfer RNA for this particular section was UAC, and now we're having UAG. That's a different anticodon. Now, I need you to understand that when we get to amino acid, there are 2 possibilities. If the anticodon, and sometimes they use the anticodon and sometimes they use the codon. If the codon or anticodon codes for the same amino acid, there won't be a change in the protein. So if the amino acid that is coded for, even if there is a change, in this case, if the amino acid is still proline, that will have no effect on protein. You will still get the same protein that is formed. Why is that? Yes, there were changes in the base, but we still had the same amino acid, proline, meaning the same protein is going to be formed. That's option number 1. But option number 2, if this base now that we have, this anticodon, codes for a different amino acid. So the initial anticodon coded for proline, and now this new anticodon codes for maybe arginine. This is supposed to be arginine. So what does that mean? An amino acid has changed. So if the amino acid changes, that means the sequence has ultimately changed. And that means we are going to form a new protein or a different protein. That is mutation. So what does this change result in? And where does the change occur? It normally occurs in the DNA bases. Now, this change may lead to the very same protein being formed, which means there was no effect. Or it can lead to a new, sometimes they say new or different protein. Now, how do we know whether the protein formed is the same or different? You look at the amino acids. If there's a change, but the amino acid remain the same, that means the protein will remain the same. But if there's a change in the bases and the amino acid name changed, that means the protein that is going to be formed will also change. Now let's go to our text. Remember, stop me any second and ask. This can be a bit too much. So please do ask me any second. Yes, my volume is on. All right. So what is this gene mutation? Remember, a gene is a section of DNA in a chromosome. And that section is basically consisting of the sequence of bases. So with gene mutation, we are speaking about the change in the sequence of nitrogenous bases. Or you can say nucleotides, the sequence of the nucleotides in DNA. This is terminology that you need to know. You need to know what gene mutation is. Now, these changes, once you see the word mutation, think change. Now, where is this change for gene mutation? This is a change in the sequence of bases, because the gene is determined by the sequence of bases. Now, this mutation is very random. Mutations are random inheritable changes in the genetic material of an organism. Now, a mutation, that change, can cause a permanent change to the DNA of the cell, because now that means if there is that permanent change because of mutation, it will change the DNA of the cell, which can ultimately change the proteins that are now formed by that particular cell. When does this mutation take place? So when exactly will this change take place? It normally takes place during DNA replication. So when DNA is making a copy of itself in that particular cell, it is possible that a base pair can be added, or a base pair can be left out, or a base pair can be doubled. So instead of having TAC, we end up having TAAC. Now that is the change. Since messenger RNA, now I want you to look at what is happening here. So I'm going to write down this. Where did the change happen? It happens in DNA when during DNA replication. Now, how will this change affect the whole process of forming a protein? It will first affect the messenger RNA. There is the point about messenger RNA. It's the same thing that I've written here. I said DNA, messenger RNA, transfer RNA, amino acid, protein. Since messenger RNA is copied from the DNA molecule during transcription, a change in the DNA will result in the change in the messenger RNA. So a change in the DNA molecule will obviously result in a change in the messenger RNA. And remember, we get the anticodons of the transfer RNA from looking at the codons of the messenger RNA. So if there's a change now in the messenger RNA, this is the point now, there will also be a change in the transfer RNA. So different transfer RNA molecules carrying different amino acids will be required. Now, from transfer RNA, what do we speak about? AA, you should know by now, that's amino acid, not alcohol anonymous. I am just writing AA to simplify it. The sequence of amino acids may change. And remember, it is the sequence of amino acid, and we are looking at it amino acid by amino acid. And an amino acid is coded for by 3 bases. Now, the sequence of amino acid may change, resulting in the formation of a different protein. If the amino acid name changes for that particular section, a different protein is going to be formed. But look at this. If the same amino acid is coded for, yes, there was a change, but for that section, the same amino acid is coded for. There will be no change in the protein structure, meaning the protein will remain the same. This is the sequence that you follow. examples of mutation. This I'm just using to explain it to you, to explain mutation to you. You're not going to be given such a diagram where you have to figure out the type of gene mutation. So let's look at our diagram here. So this is a normal diagram. What are you given? You are given DNA here, and then you are given the messenger RNA, and then you are given the names of the amino acid. So DNA, messenger RNA, names of the amino acid. Now, a possible mutation that may take place, let me figure out what's happening with my slide. Okay, a possible mutation that may take place is substitution. Now, what do we mean by that? Let's look at what has happened. So let's start from these bases. We've got AAA, ATG, CTT. Okay, we are happy. But what has happened here in this particular section? So in our original DNA, we had CTT, meaning our messenger RNA was then GAA messenger RNA codons, which then coded for amino acid glue. It could be glutamine. Now, what did we see happen here? So if you look at the very same region, we are no longer having a C as our first base in this triplet. We are now having a T. So instead of having CTT, we are now having a TTT, meaning there was substitution. C then got substituted by T. Now this changed the bases in that codon. Instead of GAA, we then got AAA. Let's look at the amino acid. The amino acid changed. We initially had glutamine that was coded for by GAA. Now, since we have AAA, it's coding for lysine amino acid. Now, this tells us that it will result in a new protein that will be formed. Same with insertion. Insertion, there's just the base that got inserted. So if we look at this triplet here, it was CTT. This was CTT. Then there is a base that got inserted before C. We then ended up changing the whole sequence of bases. We are reading the triplet from here, CCT, which changed the amino acid. Same here, deletion. There is a base that got deleted. Remember I said, I am just giving you this as an example to see what is happening, but they don't normally assess it like this to say substitution, insertion, deletion, but they do explain the change that happened. So you don't have to be spending now 3 hours on this slide trying to figure it out. How is mutation assessed in a test? Now, I need you to make a note that mutation is assessed in application. You see what I've written, showing you about the order of the molecules that are formed from DNA, messenger RNA, transfer RNA, amino acid, protein. You follow that order, but then you apply what you are given in a question. So you will come across, two types of questions about DNA mutation, which affect proteins. There is a general question, and then there is a specific example. I will tell you now, they are no longer doing general questions. So I'm going to give you the general order, but you need to use that to apply whatever example that you are given in a test. So what is the order? The order is what I've written, and that's all you need to write. A change will occur in DNA. What exactly in DNA? It will be the change in the nucleotide or in the sequence of bases. And when that change occurs, it will obviously affect the messenger RNA, and then it will affect the transfer RNA anticodons, and then affect the amino acid, and will ultimately affect the protein. That is the generic answer. Now, if we're to look at this, you need to use the generic words, exactly what I've written here, in order for you to use this example and apply that generic statement. What do I mean by this? Let's look at this question. So the information below shows the sequence of a protein's 6 amino acids. So a protein is formed, and remember we are having a minimum of 50 amino acids that will need to be bonded together by peptide bonds in order to form a protein. But now it's not possible for us to show you all 50 or even 350 amino acids that make up a protein. So they've decided to show you only 6. Now, these are proteins that are found in the saliva of Angela and Brian. Then they tell you what happened. A mutation caused a change in Brian's sequence. We are going to focus on Brian. A mutation caused a change in Brian's sequence of amino acids. The amino acids are sequenced from left to right. Now, the expectation in terms of the sequence that are found in saliva of the two people here should be this: glycine, valine, histidine, proline, isoleucine, and tyrosine. But now, what has happened here? They are telling us that there is a mutation that occurred in Brian's sequence of amino acid. Let's figure it out. So glycine, glycine, that means it was fine here. Valine, valine, it was fine here. Histidine, histidine, it was fine here. Proline, valine, this is different. Now, since we are having a protein for the saliva, or for, yes, found in the saliva, the amino acid sequence should be the same for both Angela and Brian. But what has happened here? There is an amino acid that is different. And the rest, iso, tyro, tyro. So that means the mutation occurred here. Let's look at the question. Describe how a mutation caused a change to Brian's amino acid sequence for 4 marks. You're not going to skip that 4 marks. Describe how a mutation, there's the keyword, caused a change to Brian's amino acid. What happened that led to Brian's amino acid sequence changing? Where are we going back to? We are going back to this. This means that there was a change that occurred in the nitrogenous bases or nucleotides of a DNA molecule. And since the messenger RNA copied from the DNA molecule during transcription, this will result in the change in messenger RNA codons. Even if you don't phrase it like that, mutation occurred in DNA nitrogenous bases, which resulted in a change in messenger RNA codons, which resulted in a change in transfer RNA molecules or anticodons, which resulted in a different amino acid being formed. Now, you're not going to just say different amino acid being formed. Which amino acid? Valine. It was supposed to be proline, but valine is the one that got formed here. Now, since there is a different amino acid being formed, that means there will be a different protein that is being formed. That's 4 marks. So what did I say? So I said there was a change in the DNA. Now, in this case, this is still simple because they didn't give us the basis change. So we are still mixing generic and application. So there was a change in DNA nucleotides, which led to a change in the messenger RNA codons, which then led, remember you are writing sentences, full sentences on your side, which then led to the change. The change in the transfer RNA anticodons, which led to a different amino acid that is being formed. Which amino acid? We mention it, which led to a different amino acid, which is valine. Obviously, if there is a different amino acid formed, this will lead to a different amino acid, leading to a different, not different amino acid, leading to a different protein that is being formed. There's your 4 marks. There is your 4 marks. If we answer the last question, I'm going to give you guys 2 minutes to find out the answer for the last question. It's just 1 mark. How many messenger RNA nucleotides would be involved in forming the portion of the protein shown for Angela in the table above? I want you to think about it. And then I'm going to ask a few people to raise their hands so that I can choose who can answer for us. They're asking you how many messenger RNA nucleotides would be involved in forming the portion of the protein shown for Angela in the table above. Please do raise your hand if you would like to answer so that I can just select Or if there are no hands, I'm going to call out people's names. I don't see any hands. I don't see any hands. Guys, are you there? You're too quiet. Are you there? Hi. Can anyone just unmute and give me an answer then? Okay, Lwazi. Lwazi, please do give us an answer. How many messenger RNA nucleotides?
6.
How many?
6.
6. Okay, how did you get 6?
Counted the number of amino acids.
You counted the number of amino acids. Okay, so an amino acid is, just tell me this. An amino acid is coded for by how many bases?
3.
By how many, Lwazi?
3.
Thank you. That's the correct answer. 3 bases. How many amino acids do you see in Angela's sequence there?
6.
6. So what are we going to do with that 3 and the 6?
Times it by 3, which is
Thank
18.
you. Which is what? 18. So remember, guys, an amino acid is coded for. I like how your initial answer was incorrect, but with the guidance, you are able to see, okay, there is one amino acid, and one amino acid is coded for by 3 bases. And they are asking us for the nucleotides. So we are going to count the amino acid 1, 2, 3, 4, 5, 6. And each amino acid is coded for by 3 bases. So you multiply that by 3. So that will be 6 multiplied by 3, 6, 12, 18. You get 18 nucleotides. I hope that is clear. If you have a question, guys, please remember to stop me any second. Ma'am, re-explain. We don't understand something so that I know what to do. The base is glycine. Glycine will be coded for, I'm just creating my codons here. Maybe UAG. So this one amino acid is made up of 3 bases. Each base is obviously in a nucleotide because one nucleotide is made up of one base. So same with valine, maybe valine is AAA, then this one is CGC, then this one is GG. I'm sure you get what is happening. So it will be 3, 6, 9, 12, 15, 18. That's how you get it. Okay, let's look at this one. The following sequence represents a part of a nitrogenous base sequence of a DNA molecule. You are having 3 triplets. When they say triplets, it's in a DNA molecule, but they've already told us. So it's triplet 1 and triplet 2 and triplet 3. Please, let's get ready to do this together, guys. Please, please, please. Okay, let me zoom in a little bit. Okay, so write down the base sequence of the anticodon triplet 1 shown above. So just 30 seconds to figure this one out. Write down the base sequence of the anticodon of triplet 1. There is triplet 1. They want the anticodon. Remember, when they say anticodon, they are speaking about bases in a transfer RNA. We can use our order of molecules. We move from DNA in order for us to form messenger RNA, in order for us to get transfer RNA, in order for us to get amino acids, and in order for us to form a protein. I need someone to give me the anticodons. Amanda? Amanda, please give me the anticodons. Okay, Jonita? Jonita, please give us the anticodons. Okay, guys, I'm not moving. Anyone can unmute and give us the anticodon. I'm not moving. If people are quiet, I'm not giving everyone answers.
I don't know if it's right. Is it UAC?
Okay, let me go back to my notes. Yes? I didn't hear.
UAC.
UAC, that is correct. So the anticodon here will be UAC. How did you get UAC, Renee?
I did my mRNA, which would be AUG, and then I went to my tRNA, which is AUC.
Thank you. Triplet 1, the basis was TAC, the messenger RNA AUG. Then you get your transfer RNA, which is UAC. Remember, they gave you this and they were asking you for this. So you had to work out messenger RNA and then transfer RNA. All right. Then the table below shows the amino acids that correspond with different messenger RNA codons. Give the correct sequence of amino acids for the DNA triplets 1 to 3. Our goal is to answer the mutation questions, but obviously we need to answer all the questions. The main goal is to answer this one. So give the correct sequence of amino acids for DNA triplets 1 to 3. So you are going to use this table over here in order to get your sequence of amino acids. Okay, anyone with an answer? Please do let us know. When you have an answer, guys, please do unmute and tell us. I'm not moving up until I get answers. If it means we're going to be doing this for 2 hours, so be it.
I think it's arginine.
Okay, you're saying it's arginine?
Yes.
Mm-hmm.
And then the next one, oh, sorry, I started from the wrong one. The first one is methionine,
Methionine.
and then it's
Arginine.
arginine, and then the last one is glycine.
Glycine. How do we figure this out? We get the messenger RNA first. So we had TAC. So for messenger RNA, this will be AUG. Okay, there is AUG, hence the first amino acid, methionine. Then the next triplet is TCT. We are going to take it to messenger RNA first because that's what we have on the table. And then we get AGA. So the second one will be arginine because you go to the table, simple stuff. And then the last triplet is CCA. You take it to messenger RNA, which will become GGU. There's GGU glycine. You get 2 marks for that. Yeah, you get 2 marks for that and write them in the correct sequence. So for you to get that full 2 marks, they have to be in their correct sequence. If the sequence is messed up, you actually get 0. There's no 1 mark. Then there is mutation. During DNA replication, a mutation occurred in triplet 1. I'm going to scroll up a little bit. During DNA replication, they're telling you a mutation, meaning a change, occurred in triplet 1, resulting, they're telling you what's happening, resulting in C being replaced by G. For 3 marks, describe how this mutation will affect the structure of the protein that is formed. They are asking you, is the protein that is going to be formed the same or different? That is the question. How are we going to figure this out? We are going to follow this sequence over here. I spoke about mutation. But now, we are not going to be generic. We're not going to say a change in the DNA nucleotides results in a change in messenger RNA codons, resulting in a change in transfer RNA anticodons, resulting in a new amino acid. Wrong, 0. You are going to apply what you have been given. Now, in order for you to get the 3 marks, you need to figure out what amino acid was formed first when we had triplet 1 as TAC. And then you do exactly what you are told in the question, whereby C got replaced by G, and then you figure out the amino acid that is being formed there. Then you should be able to decide, using the names of the amino acid, whether the protein that is formed was the same or different. Let's do it together. We're not skipping 3 marks and 4 marks questions. We are answering them. This is not a common knowledge question whereby you are just going to figure out what to say. We are going to follow the rules. So they've told us what to work with, and we're working with triplet 1. So we are going to follow the base and always write them on the side for you. These things, you are writing them for you. You write this in a test, they're just going to mark you wrong, because what is that? So you write them on the question paper somewhere for you to remember, okay, the base change in DNA, which means the next molecule that will be affected is messenger RNA. The next one is tRNA. So I'm going to repeat that. I'm just writing it this side because there's space. So DNA. Let's remind ourselves, which triplet are they speaking about? That is TAC. Now, for TAC, our messenger RNA messenger RNA codon, they would be AUG. Remember what I said? We need to figure out the amino acid that got formed first, right? So here, because in our table, I know the next one is transfer RNA, and I'm hoping I don't confuse you. If you want to include transfer RNA, you can. It's fine. But for this particular question, they are going to, or they have used the messenger RNA codons to get the name of the amino acid. And the name of the amino acid that we have for AUG is methionine. That's what we have. Now, if you include transfer RNA here and you do your anticodon and then you get UAC, you included this side, you're not going to be penalized. That is still correct, right? But now they've skipped a stage here. Then, what is the name of the amino acid that is being formed? Where are you going to get the name? From the table. Your messenger RNA codon is AUG. There is the amino acid formed, methionine. So this is you figuring it out on the side. Okay, then remember there's a change that happened. There was mutation that happened. What did they say? TAC, the C got replaced by G. I am going to change my color just to show the difference here. So remember you have TAC, but then they're saying the C got replaced by a G. That means the base that you end up having is TAG. I hope we're still following. So if now in the DNA triplet, the change obviously made sure that you are no longer having the C, you are having a new base. So what does that result in terms of messenger RNA codons? So A is going to pair with T. We are forming our codons now, and A will be paired with U, and G will be paired with C. Let's look for our amino acid. You then take your AUC and you look for it in the amino acid table. There is AUC. What is the name of the amino acid formed now? Is the isoleucine. Yes. Please do unmute if you have to unmute. I think it makes me feel better, because when I'm just talking alone, oh my goodness, I don't know what's happening on the other side. And this is stressing me out, because no one in this group should be losing these 3-4 marks. No one. I saw. Okay. I then need you guys to tell me, is the protein that is going to be formed now, will it be the same protein or will it be a different protein? Please tell me.
It would be different.
Thank you so much. A different protein is going to be formed. This is an application question. Now, how do you phrase it? Fun fact, everything that I've written here should be part of your answer. You start by telling us what happened with the mutation. T in the DNA nucleotides. I'm just not going to be able to write the whole thing. But what I've written here is exactly what needs to be in your answer. Then you phrase it in a sentence. In a DNA nucleotide, which had TAC, A got replaced by G. That means we then ended up having TAG. 1 mark for that. And then, which led to the messenger RNA codon being AUG. AUC, can't see that's blue. Which then changed the amino acid that got formed from methionine to isoleucine. Or even here, you can say the triplet TAC, because of mutation, then became TAG, which caused the change in messenger RNA codon from, yes, I think that is important, from this to that. You are showing us the change from AUG to AUC, which led to a change in the amino acid from methionine to isoleucine. Fun fact, well, it's not fun, this one. If you mess up the spelling of the amino acids, wrong. Why? Because you are given the amino acids. So it's a copy and paste. You can't mess up the spelling there. So then you tell us, resulting in a different protein formed. It's either you say different or new protein formed. That's your 3 marks. We are not skipping these questions. We are answering them, and correctly so. That's what we want. I'm happy to take questions, guys, or a comment or something, please. Something, please. Okay. We're going to move. I'm going to skip all of this and
Sorry.
go to, yes?
Sorry, ma'am. Can you please repeat the DNA part?
Let me scroll up. Which part exactly?
When you explained the mutation for question 2.1.2b.
Okay. Mutation occurred in triplet 1, where C got replaced by G, or you say where triplet TAC became TAG.
Okay,
Or it
ma'am,
changed
thank you.
from TAC to TAG.
All right, ma'am, I understand. Thank you.
Is it sorted? You got the rest of the points?
Yes, thank
Okay,
you.
thank you. All right, I'm gonna skip this. These are basic activities. I'm now going to go to, what is this? Profiling. We're gonna do profiling. But these activities are also important, funny enough, but yeah. So I'll send the memos to the group. Okay, we're here. DNA profiling. Okay, thumbs up to show me that you are still here and you are listening and you are active. Please, guys, thumbs up, everyone. Yes, 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 yes. Yes, please. Okay, thank you. Let's continue. So this is a very small section of DNA, and it is the last part of DNA. We call it DNA profiling. Please cross out this word. It was accepted in the past, but they're no longer accepting it now. So when you think of DNA profiling, you actually need to think of certain situations whereby I think we've watched a show whereby a couple, or rather a woman and a man with the child that go to a doctor, and then they take their DNA to determine if the guy is the father or not. That's the mindset. That's the DNA profiling business. Or in a crime scene, then the forensic people will come and collect the DNA that is left in the scene, and then go to the labs and figure out who are the possible suspects, victim, and all of that. That is what we are speaking about. We are not speaking about, when you go to the bank and you open your bank account or whatever, you use your fingerprint DNA. That is not what we are speaking about. So you use DNA profiling, whereby they will collect a sample and then get your DNA from that sample. It's not this fingerprint business. Now, this section is very basic, and you will see it is easy. This is easy 6 marks that you can get. What is a DNA profile? A DNA profile is a pattern of black bars. So from the DNA that they collect from your sample, they're going to run it through specific biological tests. When they run it through the specific biological test, it will then end up forming bars there or lines. You will see them as lines. Think of a barcode. Everything that we buy has a barcode, and that barcode is normally specific to that particular product. So now that means those bars or lines that they are able to read after they put your DNA through that biological test will be unique to you. So each and every individual should have their unique DNA. The only people that have the same DNA is identical twins. So if you're an identical twin and they run your DNA and they're trying to figure out who committed a crime, it's going to be difficult for them to prove which twin it is because their DNA is the same. They are from the same sperm cell and egg cell, hence their DNA is the same. Now, the chemical structure of DNA is the same for everyone. The only thing that is different between my DNA and yours is the sequence of bases. That's it. Then I think, okay, I'll still read this. Millions of base pairs exist in each individual's genome. That is your full set of your DNA. But it is very expensive and time consuming to identify all the bases. So when scientists do this DNA profiling, they are basically using a shorter method in order to identify people. So they will just get a sample and then get your DNA from that particular section, which should be unique to you. So they can use DNA profiling process to identify individuals, to determine genetic relationships between people, siblings, and all of that. Now, in this method, we are looking at the patterns of DNA. That's it, nothing more. The bands on the DNA profile represent the different size of DNA fragments, and your size of DNA fragments should be unique to you for that particular part. So in this case, we shouldn't stress about how exactly they do this process and whatever. We should just know how to analyze the DNA profile. That's it. So I'm going to skip this part. This one is important. I'm going to skip this section only. So what they're going to do, they're going to analyze the DNA for the unique sequences, but we're not going to see A, C, G, T here. They will just use those bars, which should be unique to you. And that sequence should be able to identify individuals and genetic relationships. I think I've mentioned that, siblings, parents, paternity, and all of that. Every person, I've mentioned this, except identical twins, they have their own DNA profile. Every person should have their own unique DNA. Even if you are siblings, different DNA. Now, the exception is identical twins. But remember, with twins, we've got two types of twins. We've got identical twins, and these ones will have the same DNA. But we also have another set of twins, which we call fraternal twins. And fraternal twins have a different DNA. So when we speak about twins, you need to be specific about which ones you're talking about. Identical twins will have the same DNA, but fraternal twins will have different DNA. Fraternal means there were two different sperm cells and two different egg cells. If the twins are of a different gender, a male and a female, definitely they're fraternal, but it is possible to have twins that are of the same gender, but they look very different. They look like siblings. They're still considered fraternal twins. So in DNA, if you just say twins, we mark you wrong because we don't know which twins you're speaking about. Now, how are they going to get this DNA that we're speaking about? They are going to collect a DNA sample. So they need a sample, which is obviously containing your cells, and then from that cell, they'll get the DNA. They will then perform a test. And how do they need to obtain this? They can get it from the blood. They can get it from your hair follicle, saliva, semen, skin, or any cell, because any cell that you have in your body will have your DNA. Now, they will need two samples in order for them to compare. So if we are having this person's DNA profile and this person's DNA profile. We should be able to have 2 in order for us to compare. But when we do paternity DNA profiling, there will need to be 3. We need the profile of the child, of the mother, and the possible father. Then we are able to determine if the guy is the father or not. This slide is important, and this is 3 marks. 3 marks. So just remember 2 or 3 here to be safe. 2 or 3. Now, these are points talking about the uses of DNA profiling. It should be profiling here. Profiling. Don't write fingerprinting at all. So we use DNA profiling to do paternity testing by just writing paternity testing, a tick. So paternity testing, this is when they determine the father of a child. Then DNA profiling can also be used in forensics plus, but please note for this point, you can't just write forensics. They won't give you a mark. You say forensics to identify organisms from their remains, then that's a mark. So if they find a person that has obviously unfortunately died, they're able to run their DNA quickly and then identify that person. So for this one, you will need to say forensic and then you add the point. If you just say forensics, no marks. Also, another use in forensics to identify the crime suspects in forensic investigation. A crime has been committed. Forensic people will go there and collect samples in order for them to run through a biological test, biochemical test, to then get the DNA. In forensics, they can also use DNA profiling to trace missing persons. Please see where the tick is at. Now, we can also use DNA profiling to identify genetic disorders, to identify family relationships, to match tissue types for organ transplants or organ donation. I'm not going to explain everything else because they don't need you to explain more. So you just need to write what I've underlined and the tick. Remember 2 or 3 here, that's it. That's 3 marks that I need everyone to have. This is another slide. I need you to also remember a minimum of 3 points here. You don't need to remember everything else. What are some of the limitations with DNA profiling? So yes, we can get forensic people to collect samples from a crime scene. Yes, we can get forensic people, not forensic people, lab people to then determine paternity for a particular child. But there are limitations to that. Yes, the results from DNA profiling are known to be reliable. So if you do the process, we should be able to identify whether this guy is the father or not, whether this person is a possible suspect or not. However, there are limitations. Now, the limitation because the interpretation and the comparison of the profiles should be, however, approached with caution, but we are speaking about human beings that are running this. So they are interpreted by human beings. The machines are going to do everything, but you need a human being to say, yes, you are the father. This is what we're able to find. Or yes, you are not the father. This is what we're able to find. I need you to just remember 3 points here. 1 point of limitation is human error. By writing human error, tick. So if now I'm the one that did the test and then I'm seeing the bars, then I'm having this woman and the man and the child, but normally the child is not there. So I tell this guy, hey, you are the father, only to find out that I misunderstood the lines or I didn't interpret the lines correctly. That is an error on my side. There are short segments. If small DNA is used, I would say ignore this one. I would also say consider this one. Simple contamination with another DNA that is possible. So if then there is a sample that has been collected, but that sample was mixed up with another sample, then the DNA that will be read there will probably be incorrect. So remember this one also. I'm just trying to choose the easy ones. Okay, DNA decay, that particular section of DNA, prejudice, that is also possible here. You can also speak about bribes. People can be bribed to interpret the results incorrectly. But yeah, let me go through everything. Uniqueness, there's a very small chance, but about 1 in a billion of 2 different people having the same DNA. It is expensive. DNA profiling is expensive and therefore not readily accessible. Obviously, people need to pay, so there's other limitations to it. But remember, like I said, 2 or 3 things. Human error is normally very common. Contamination, DNA, decay, prejudice. Even with this one, just one word, you get a mark. You don't even have to elaborate. Now, what is expected from us? Yes, the limitations and the uses we need to know. We need to know how to interpret a DNA profile. This is a DNA profile. Now, there's rules that you need to follow when you interpret it so that you can give the correct answer. What do we look at? We look at the position of the DNA bands. I need you to make a note of this. We call these lines, you see these lines that we are seeing here? We call them DNA bars or DNA bands, nothing else. No other word is going to be accepted. Not lines, not black lines, not sections, nothing else. They're not going to take it. And you have to include the word DNA. You can't just say bars or bands. You're not going to be given a mark. Now, what do we do here? We compare the position of these DNA bars and DNA bands. So when we look at it, so if, for example, I'm going to change my color so that this can be visible. Okay, so if we look at this DNA band over here, this first one, we are going to compare it with the same position of the suspect. In this case, we are having DNA profile that was collected as evidence, and then the possible suspects here. So we are going to use this band over here and check if this band is available in suspect 1 in the same position. If it's not available, we'll move to the second suspect and then to the third suspect. Now, in order for the possible suspect to be the one where we say you committed a crime or whatever, the DNA profile needs to 100% match. We do not speak, yes?
No, ma'am, I was agreeing to what you're saying.
Oh, okay. Thank you so much. That is very important. It's not a matter of, okay, suspect whoever is okay, let's say choose suspect 2. For example, I haven't looked at the bands, but we're going to look at them now. Suspect 2 is the one who committed a crime because suspect 2 has more DNA bands that match the ones that we found in the evidence. Wrong. Not about more DNA bands. It has to be 100% of the bands. Now, if all the bands in 2 samples are in the same position, check what are we talking about here? All, not some, not most of the bands. If you say some of the bands, most, wrong. Then the DNA samples are a match if all the bands in the 2 samples are in the same position. Now, let's look at our example and then explain how we read this. Remember, we're saying this is the DNA profile that was then found as evidence, right? And these are the possible suspects, suspect 1, 2, and 3. Me and you need to figure out which suspect are we saying committed a crime. So what are we going to do? We focus, I just don't have a ruler. I'm not sure how I'm going to show you this, but we focus on the first band in the evidence. We're not going to focus on the suspects. Our key focus is the bands in the evidence. And then we look for the bands in that same position in the suspects. So the first one is this one here. Do we find it in suspect 1 in the same position? No. Do we find it in suspect 2? Yes. So if you are using a highlighter, you can highlight this DNA band. What about in suspect 3? We don't have any band in that position. Now, already this eliminates suspect 1 and suspect 2 before we even continue. Why? Because if there is no band in suspect 1 in this position, and there is no band in suspect 3 in this position, remember, we need all the bands to match. already there are bands that are not matching in 1 and 2. So our key suspect is this gentleman over here. I'm biased. Why am I saying it's a gentleman? It could be a female. We don't know. So in this example, the DNA bands in the evidence sample and those of suspect 2 are in the same position. Therefore, they are a match. So if they were to ask you to give a reason, why are you saying suspect 2 is the one who committed a crime? This is your answer. The DNA bands in the evidence sample and those of suspect 2 are in the same position. Therefore, they are a match. Even this therefore is not needed. The tick will be there. Remember, DNA bands, DNA bars. If you say the lines are in the same position, wrong. We're not going to lose that 2 marks. So there's normally 2 examples, right? So one will be for the crime and the suspects, and the other is normally for paternity. Let's look at this. Now, in paternity testing, the child will have some bands in the same position as the mother. Check the words that we have. Remember, in the crime scene business, we said all the bands. With paternity, some of the bands will need to be in the same position as the mother, and some of the bands will need to be in the same position as the father. What is the idea here? The DNA that a child will have would have been inherited from the father and from the mother. But let's understand this. A child inherits 50% of their DNA from the father and 50% of their DNA from the mother. That means if we are having 10 bars, 5 of those bars should be accounted for by the mother, and 5 of those bars should be accounted for by the father. Why is that? Because we said 50-50. If you are shown the band, and I'm not saying we now should count the bands and say, okay, there's 18 of them and 9. That's not what I'm saying. But I'm saying 50% of the DNA that the child will have would have been from the father, and the other 50% will be from the mother. Now, what is our focus when we are doing a paternity DNA profiling? Our focus is the child. Let's remember, when we're looking at the DNA profile of the suspects and evidence, our focus was the evidence. We were looking at the evidence and then trying to figure out which suspect has bars in the same position. Now, when we do paternity, our main focus will be the child. And what exactly are we trying to find here? We need each band from the child to be attributed to either the mother or the father. That band here that is representing a fragment of DNA should either be from the mother or the father. It can't be from both. Now, in a case where you are having a band that is in the same position as the mother and the father, the rule here is to start with the mother. If the mother has that band in the same position, you account it to the mother. Now, yes, you can argue why is that, it could be the father and whatever, but this is the rule of how we are reading this. Now, look for bands that cannot be accounted for in the mother's profile, and then compare these bands to the potential father's bands. That's exactly what I've said. Now, let us look. This is the child, and this is the mother, and there's 3 possible dads here. Okay. So remember what I said, we are not going to come to the mother and figure out the bands. We are not going to the 3 possible dads. We go to the child because we need to account, okay, there's a band here. Does it belong to the mother or to the father? That's why we are focusing on the child. So this is the band over here. Let's account for it. Where do we start? We start with the mother, always. We look for the same band in the same position from the mother first. Do we have this band in the mother? No, it's not there. It should be over here. It's not there. So since it's not there in the mother, we are going to look for that band in the possible father. Do we get this band? You use a ruler here. I just don't have a ruler with me. Do we get this DNA band in dead 1? No, it's not there. Do we get it in dead 2? Yes, in the same position.
Yes.
Thank you. Then in dead 3, there it is.
Yes.
If we have to highlight, you highlight. Right. Each band has to be accounted for. But I'm sure you can see what's happening. We can now eliminate dead 1. Why are we eliminating dead 1? Because there was no DNA band in the same position, and it needs to be accounted for by the father because it's not there in the mother. So already dead 1, bye. You are not one of our possible fathers here anymore. So now we are left with 2 and 3. We don't leave it there. We move. We go to the second DNA band. Do we get this band from the mother's DNA profile? Yes or no?
Yes.
So we
Yes.
stop here. We found it in the mother. Do we need to look for it from the father's side?
Yes.
No, we don't. We found it.
don't.
Remember, a band needs to be accounted for by either the mother or the father. So if we already have it in the mother, we're not going to look for it in the father.
Oh, okay.
Yes, that's how we read it. Because what are we basically saying? We're saying this section of DNA was inherited from the mother.
From the mother.
Because we can't have the same band being accounted for by both parents. That's not one of our rules. Okay, so that means this is accounted for. Then we go to this band. Remember, where do we start? With the mother. So is this band found in the mother's profile? Yes.
Yes.
There it is. So do we look for this band in the father, in one of the fathers?
No.
No, we don't. So I don't want you to be confused and say, okay, this band is in the same line as this one in dead 1. We already found it in the mother. We don't even look at what is happening with the dads. Then what about this band over here? Do we find it in dead 1? Yes, we do. But remember, dead 1 was eliminated already. You can still highlight dead 1 if you want. Let's see. And then we're not finding it in dead 2. And then in dead 3, we are finding it. There it is. Remember, we need to read all the bands. We can't just make a decision by 1 or 2 bands. We need to read all the bands from the child. Then what about this one? We're not finding it in the mother. We start with the mother. It's not there. So that means we go to the possible dads. In dead 1, it's not there. In dead 2, there it is. Am I making a mistake here? I think I made a mistake somewhere, but it's fine. I'll figure it out. This one, because I don't have a ruler. I'm trying to. Please tell me if I'm making a mistake because I don't have a ruler here. I don't want to say they're in the same position while they're not.
It's the same position.
Is it the same position?
Yes, ma'am.
Oh, okay. Thank you. Then what about this one? This next one over here. Okay.
Dead number 3 also
Dead
has.
number 3. Thank you. I'm struggling. I'm trying to use this as my ruler and it's messing me up. Okay. Then the last one, let's look at the last one. We are finding it in the mother. Now we need to decide which one is the dead. How are we going to decide? Remember, we should have highlighted, if you did highlight, then you're going to count the number of bands that are matched. In dead 2, there were 2 bands that were matched. In dead 3, there were 3 bands. So between the 2, who had more bands? It was dead 3 because the other bands were accounted for by the mother. So that means dead 3 is the father. Is it clear, guys? Thumbs up if it's clear. Because I'm hearing 2 voices only. Thumbs up if it's clear or something. Thank you so much. This is how we read this. This is 4 marks that we cannot afford to lose. Okay? All right. This one is for you. This one is for you. Two. You know what? I'm going to be stingy. 1 minute. 1 minute.
Offspring.
30 seconds. Okay, anyone with the answer? Identify the male who is the father of the offspring. There is the offspring. Offspring is the child. Which one is
Is
the
it
father?
male too?
We think male too.
Male.
Male too.
Male
Yes, male
too.
too.
Yay! It's definitely male too. I'm happy. You see, now I'm back to being happy. Okay, we are back to the suspects. Tell me which one committed a crime. Ah, this one is easy. Here, you're not even using a ruler. Which suspect? Suspect 1, 2, or 3?
Two.
Definitely suspect 2. Then I need someone to give me a reason for 2 marks. Why are you saying it's suspect 2 for 2 marks? Anyone?
Because the first DNA band matched with the crime scene of the DNA band.
Because the what?
Because the first DNA band for suspect 2 matched with the crime scene, so there's no need to check, and the rest didn't.
Okay. The marking here is very strict. We don't speak.
I would
Adria?
say all the bars matches the suspect, the evidence.
Thank you. Guys, here they are too strict. As much as how we analyze it to say, okay, we went to the evidence and then the suspect, the first DNA band was there and the rest were not there, blah, blah, blah. Correct in how we analyze it. But when we are asked to provide a reason, you speak about all the DNA bands. All the DNA bands in the evidence sample match those of suspect. I don't remember which suspect it was now. Now you have to use the words that you are given here. All the DNA bands from the crime scene match those of suspect 2. That's 2 marks. So how we analyze it, that is our rule. But your answer, all the DNA bands, this is your reason. All the DNA bands or bars, it's supposed to be bands, my pen is acting up. From where? From the crime scene. Remember in our other example, they'd written evidence, so we can't write evidence. We take the words that we are given here. From the crime scene, match those of suspect 2. I need us to get these marks. Suspect 2, that's how they want it. So if they had given you the names of the suspect, I'm going to use male names, I'm not sure why, but if they had said Vuyo, Cole, and Zugo. So if this was Cole, you will need to mention Cole's name. All the DNA bands from the crime scene match those of Cole. You have to be specific. Okay. There's another one. There's a blood stain, and they're trying to find out whose blood stain is that. This is easy. Easy peasy. Is it Lisa's blood stain, guys? Is it Lisa, Sue, Bob, or John?
It's John's.
It's done.
Thank you. It's definitely John. Why are we saying it's John? For 2 marks. I don't want the same people speaking all the time, guys. I need everyone to speak. So for 2 marks, tell me why you're saying it's John's blood stain. Anyone? Anyone, please? Okay, for this one.
Because all the DNA bars in John's matches the blood stains found on the crime scene.
Okay. Yes, that is correct. Just trying to remember, did you mention the word blood stain somewhere in your answer?
Yes,
Okay.
I did.
All, thank you. All the DNA bars found in the blood stain match those of John, or all the DNA bars of John match those found in the blood stain. I need blood stain, I need John, I need all the DNA bars. Ooh, okay, I'm happy. Okay, let's find out who is the father of child 4 and yes? Oh, okay, I thought there was a question. So this is what we have here. You are having a mother and a male 1 and a male 2. So there's 2 different fathers for the kids here. So you need to tell me who's the dad for number 4 and who's the dad for number 5. So there's 2 different fathers. So male 1 needs to be accounted for, male 2 needs to be accounted for also. And this is the mother. So it's 2 different fathers, same mother, 2 kids. Okay, who's the father for child number 4? Is it male 1 or male 2?
Is it male too?
For child number 4? It's
Yes.
not male 2. It should be 1.
Oh,
Yeah.
okay, but I also didn't count fully, so
And you didn't look at, but also you see this one is confusing because you now have to go to the child and then look at male 1 and 2, or well, child, mother, then male 1 and 2, if you're not finding the bar on the mother. So it's a bit confusing, but yeah, that should be male 1 and that should be male 2. Okay. Oh, okay. Let's answer this one, guys. Thought I was going to be able to push and get other things. What am I doing? Done, but okay, this is the question. So I'll give you guys 5 minutes for this one. Then we mark it together. Two minutes left. Can we mark, guys, or do you need 1 extra minute? Please do let me know. Can we mark thumbs up for 1 extra minute? Just unmute and say, ma'am, one or 2 extra minutes. Okay, no response. I'm guessing you guys are still writing. Can we mark, please? Please, 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 please. Okay. All right. Let's mark. Or am I rushing you guys? Or can we mark, like, can I get something? A yes or a thumbs up or something, please? Please.
Yes, ma'am, we can mark.
Thank you so much. All right. Identify the 2 biological children. Anyone? There's people that have not spoken the whole lesson, guys. Sandiswa, please.
I think ma'am Gupriya no Halai.
Priya. So you're saying Priya, no? No Hila.
It's
So you're
Hila
saying it's-
and Leo.
I said Leo.
So what did you guys say?
I said Hila and Leo.
Yeah, Hila and Leo.
Definitely it's Leo and Hila. Okay, because with Priya, we can see that even the first bar, it's not accounted for by the mother and the father. The first bar is here. So we then compare it there. There's nothing from the father, there's nothing from the mother. So Priya, we can eliminate Priya very, very easily. Okay, but it needs some getting used to. So you should be fine after we do more activities. Okay, so we've identified the 2 children, we get our 2 marks. Now explain your answer for 2 marks. I need someone to explain. Anyone? What did you say for 2 marks? You should be able to say your answer, guys, so that you check whether your answer is correct or not. Skandisa? Okay, anyone else, guys?
Ma'am, I don't know if the wording is correct, but I said majority of the DNA bands of Hila and Leo's matched the father compared to the mother. But I don't know.
Okay, say it again. The DNA bands of Hila and Leo, you said majority match the father compared to the mother.
Yes.
But you know how that answer makes it sound? It makes it sound that if we're talking about percentages, we are saying these are the kids because they have a higher percentage of DNA inherited from the father than the mother. That's how it sounds.
Oh,
You see?
wait. Okay. I
Yeah.
see.
So we also read into the answer and be like, if this is changing how it's supposed to sound, then we probably penalize you. So here you can speak about some of the DNA. We can use the word some because remember I said as much as I speak about 50 and the number, you don't look at the number because they would have just used the section. Some of the DNA of Heli and Leo are found in the same position of the mother and the father. DNA bands need to be there. Mother and father needs to be there in your answer. Heli and Leo needs to be there in your answer. Don't speak about lines. Speak about the DNA bands of the 2 kids. Some found in the mother and some found in the father. Actually, that even sounds better. The DNA bands of HeLa and Leo, some of the DNA bands of HeLa and Leo are found in the mother and some found in the father. Because some, yes, but if you're saying more, it's like, okay, cool. There's actually just a higher percentage of that. State 3 other uses of DNA profiling. We covered this. So when they said other, what we are having in the diagram is one to determine the parents. So you can't say anything about paternity here. You can speak about the tissues, you can speak about all the other points about forensics. So anything about paternity, it would not be marked for this particular question. All right, we've reached the end. Any activity in this slide, I will try and send the memo for, but this is a section where we need to get high marks. I'm going to show you last year's November question. It was in the document that I had sent. This was last year's November question, and they marked it very strict. So just give me a second. I will show it to you now. There it is. I'm not sure if you can see it on your screen. Just thumbs up if you can see the question. It should be a normal question. Let's see. It has information about sample from the crime scene, sample from the victim.
But it's a bit too small.
It's a bit too small. Okay, let me zoom it in a little bit. Is it better?
Yes.
Okay, there it is. So this, I'm sure you can see the date there. This is the date. This is last year's November question. So we need to try and do this question. What is the total? 4, 5, 6, 7, 8, 9 marks. So the diagram below shows a DNA analysis of a sample obtained at a crime scene, a sample from the victim and samples from 2 suspects. In their investigation, police conducted a comparison of the DNA to identify the criminal. 221. Name the technique used in this criminal investigation. What is this technique over here? What is this technique? Lulu? What is the technique here?
Isn't it not? It's forensic investigation.
The technique used. So this is a forensic investigation, correct? So what technique do they use in order for them to determine who is the criminal? What is this? What are we able to see here?
DNA profiling?
DNA profiling.
Thank you. DNA profiling. Okay. Which one of the suspects, 1 or 2, was at the crime scene? So I'm going to give you a
Suspect 1, ma'am.
suspect 1. Do we agree with suspect 1, guys?
Yes.
Definitely
Yes.
suspect 1, because you are going to compare the profile from the crime scene with all the suspects. So there's actually 2 suspects, not all. Suspect 1 and 2, and it's definitely suspect 1. Now, give one observable reason for your answer to question 2.2.2. Why are you saying suspect 1 is the one that was at the crime scene? And check how they phrased it. They didn't say who committed a crime. Which one of the suspects was at the crime scene? So why are you saying suspect 1 was at the crime scene? Give me a reason for 1 mark. Anyone?
The DNA banding pattern of suspect 1 matches the crime scene sample.
All. I need you to include the word all. Yes? I need your same answer, but include the word all in there.
All.
All the DNA bands.
Oh, all the DNA bands show that suspect 1 matches the crime scene sample.
Okay. All the DNA bands or bars from the crime scene or from suspect 1 matches the DNA bars found in the crime scene. All the DNA bands or bars of suspect 1 matches the DNA bands found in the crime scene. That's a mark. Okay.
Yes,
Then,
ma'am.
thank you. Give 2 possible reasons why the suspect identified in question 2.2.2, why suspect 1 might not be convicted if only this evidence is used. You need to remember one of the slides that we covered. Give 2 possible reasons why the suspect identified, which is suspect 1, might not be convicted if this is the only evidence that they use. 2 marks. What would you say?
They could be an identical twin.
Could be an identical twin. 1 mark. What's another answer? What's another option?
Can we say that they were framed?
They could be framed. That's another point. What about human error? There could be human
Yes.
error. All of
Or
them.
prejudice.
Correct. Correct. Explain. Oh, 4 marks. Explain how the technique named in question 2.2.1 is used in paternity testing. How is DNA profiling used in paternity testing? Remember what I said about paternity testing. Does anyone want to try first? Anyone who wants to try? This one was a bit complex. So how the technique DNA profiling is used in paternity testing. Let's remind ourselves. So when they test for paternity, what is the first thing that they need to do? What is the first thing that they need to do? Do they just go to the labs and run DNA testing? Is that the first thing they do? No, they need to collect
They collect the
samples.
evidence.
Samples, yes. So in this case, we're not gonna call it evidence. We normally use the word evidence if we talk about crime scene or something that happened, but they speak about samples when we speak about paternity. So the first thing would be to collect samples. Who are we collecting samples from? Tell me. Is
The
it
mother
that's the mother
and the father.
and the father? Is that it?
Oh, the child
The
or
child
children.
also. This was 1 mark. When they run this whole testing, they first had to collect samples from the mother. Actually, they were even strict. If you said mother, father, child, you were penalized. You had to say mother, possible father. Because remember, we're trying to find out if this guy is the father or not. So in this case, he is still a possible father. They had to collect samples from the mother, possible father, and the child. Then what do they do with these samples? They then extract DNA from the samples. They then run DNA profiles here. What will then they do? They will then check the DNA bands. Yes, I'm listening. You can continue, yes?
They will compare the DNA bars.
They will compare the DNA bars. And what exactly are they going to look for here? So when they compare it, what do we need to find out? What do we need to get from the bands? Is it anything to do with matching?
Yes.
Okay, what about the matching? Tell me.
They need to find out if the possible, they need to find out who is the possible father for the child.
Let me guide you. From the profile, so they will get 3 profiles. The mother's profile, the father's profile, and the child's profile. My pens are going to be the profiles. The mother's profile, the father's profile, and the child's profile. So what is the first comparison that they're going to do? Are they going to compare the mother and father's profile, or they're going to look at the mother's profile and the child's profile? Will they just look at the mother and father, or they will look at the mother and the child's profile first?
They will look at the
Mother
child.
and child.
Thank you so much. So they will need to compare DNA bands from the mother and the child. Then any band that is not accounted for by the mother, they will then look for those bands where? From the father. 4 marks. 50% of the bands should match the bands of the mother, and 50% of the bands, DNA bands, should match with the father. If that is the case, 50% matches with the mother's DNA and 50% matches with the father's DNA, then that guy is the father actually. That was 4 marks. First thing, we collect samples. We run DNA profiles. They then look for bands from the child and match them with the mother. The remaining bands are going to be matched with the father. If 50% of the bands match the mother and 50% of the DNA bands match with the father, then the guy is the father. 4 marks. That's how they do it. But if you're going to say, if most of the bands match with the possible father, then he is the father. Wrong, because we don't work on most and least. We work on 50%, 50%. Okay, I'm happy to take questions. Any question? Because I don't think we'll be able to start meiosis now. Yes?
Do they ever do like in an exam ask about like maternity testing, or is it always just paternity?
Funny enough, it's always paternity. It's
Okay.
always paternity. I haven't seen maternity unless 2026 is the year, but yeah, I haven't seen. It's always paternity. Then I guess if they are asking about maternity, if the father is the obvious one, we're trying to find the mother, we'll use the same technique. But if now we know the father, meaning the DNA bands of the child will be compared with the father first, because that's who we know, that's who I'm sure about. And then the remaining bands will be compared with the mother. So I think that's a good question. So that means we'll have to start with the father, and then the mother. All right. Okay. Anything else, guys? All right. As you can see, I mean, this is another 2 hours now. We're just doing 2 things. There is a lot to cover. I'm trying to figure out how I'm going to plan other topics. I might have to record videos and post videos online for you guys to watch, or if I'll post them on weekend or whatever. I'm going to try to figure something out. But time is not on our side. And I don't just want to rush through a section and not do the questions, because you get to understand or know whether you understand the content or not when we do the questions. So please, I'm begging you, next week, I'm going to start with meiosis. Please make sure by the time you sit in for this lesson, you have studied meiosis and you are done with it. So that when I speak about the different terminologies, you don't get confused. Please, I am begging you. Meiosis we'll cover it over 2 or 3 lessons, and then we do genetics, and genetics is long. It's 45 marks, I think. So it's probably going to drag a little bit. So I'm going to figure out how we do things, but at least I know after we do genetics, I will consider myself done with paper 2, because evolution, you can study evolution straightforward. We will do previous papers for evolution, but there won't be time for us to cover 54 marks. Evolution is covered over 4 weeks at school. So that is 4 hours a day. No, I'm lying. That is 4 hours a week. So that's 4, 8, 12, 16 hours. There's no way we'll be able to cover that. It's just a lot of work, but I will figure out what we do with evolution. So after genetics, we're now going to go to paper 1 topics. There's small topics in paper 1, but we'll probably start with the big ones. Human reproduction. is one of the big ones there, and then we take it from there. That is my plan. I'm happy to take suggestions about anything else that we can do in order to speed up the process, but I think doing content and the question papers, it works. So at least when we know we're done with the topic, we are confident to say we've covered that topic. Anything from your side, guys? Anything at all?
Ma'am, I don't know if it's wrong, but I just wanted to ask if has the other group A already done meiosis? If they have, can we watch their video just to prepare for our class next week?
Okay, I can definitely do that. If I don't send it tomorrow by 12, just send me a message in the group. So they should, so the other group, group A, we've already done 2 lessons on meiosis already. So I'm
Okay.
happy to send you the links. There will be 2 different links for those lessons. Then you can catch up. That is a good idea. So if tomorrow by 12 I haven't sent anything, just remind me on the group.
Thank you.
All right, thank you. Anything else, guys? All right. I really do appreciate you taking time to attend these lessons. And I really, 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 from the bottom of my heart, want everyone to do well. The topics are not easy. The